Hello students, in today's class we are going to learn about different stages of prokaryotic translation process. So prokaryotic translation can be briefly divided into 5 stages: activation, initiation, elongation, termination and post-translational modification. So per se, these 4 stages will represent the actual stages of the actual translation process, while post-translational modification are basically the changes that happen in the protein once the translation process is completed. So first stage, that is activation of amino acids. For this we need 20 amino acids, 20 different amino acid transfer RNA synthetases, each specific for one enzyme specific for one amino acid. 32 different transfer RNA molecules, energy in the form of ATP and Mg2 plus ions as cofactor. So the enzyme amino acid transfer RNA synthetase basically catalyzes attachment of the amino acid to the corresponding transfer RNA molecule. This reaction takes place in the cytosol, wherein we have bond formation between the amino acid and the transfer RNA molecule. Energy is hydrolyzed during the process and it requires Mg2 plus ions as cofactors. Um, the initiating amino acid in case of prokaryotes is methionine residue that is further modified by a formylation reaction. So, the first step involves bond formation between methionine amino acid and transfer RNA molecule. The transfer RNA molecule for methionine is known as transfer RNA methionine. Uh, once, in case of initiating methionine amino acid, the first step is similar, where we have bond formation between the transfer RNA and the methionine amino acid. Also, the transfer RNA that binds the initiating methionine amino acid is known as or referred to as transfer RNA FMET. After bonding between the methionine and the transfer RNA molecule, in a second step that is catalyzed by enzyme transformylase, uh, in which N-formyl tetrahydrofolate acts as a formyl group donor, there is formylation of the methionine amino acid. Coming to the initiation stage of protein synthesis. So once we have all the pool of activated amino acids, that is amino acids that are bonded with their transfer RNA molecule, the initiation stage begins. So for initiation, we require 30S ribosome that is separated from the 50S ribosomal subunit. So we need both subunit, the messenger RNA sequence, initiating formaldehyde methionine transfer RNA, 3 initiation factors 1, 2, and 3, energy in the form of GTP and Mg2 plus ions as cofactors. The initiation stage will begin at the AUG codon. It is towards the 5' end of the messenger RNA molecule that is being translated. It is not the first 3 nucleotides on the messenger RNA. And also this AUG codon is going to determine the reading frame. Now, in case of prokaryotes, there are a conserved region of about 7 nucleotides present upstream of the initiating AUG codon. This region is known as Shine-Dalgarno sequence and it reads AGG in 5' to 3' direction. The sequence of the Shine-Dalgarno region is complementary to the sequence of the 16S ribosomal RNA of the 30S ribosomes. Thus, there is a specific base pairing that takes place between the complementary sequences that stabilizes the binding of the messenger RNA on the 30S small ribosome subunit so that the AUG start codon is present at the proper location on the ribosome. This slide discusses about the three possible reading frames in a sequence. Uh, so, in a double-stranded DNA, there are actually 6 different possible reading frames because there are 2 strands of DNA, and on each strand of DNA, there are 3 possible reading frames. So, for example, if you start from adenine, you start reading the sequence from adenine, then AGU will form serine amino acid. And then automatically the next 3 bases will form the or encode for the next amino acid and so on. However, if we consider the second reading frame, which is starting from guanine instead of adenine, then GUC will form the first codon, and hence the reading frame has changed. Similarly, in the third reading frame, the first codon can be maybe UCU, which encodes again for serine, and hence the entire reading frame has again changed. So in one strand of DNA, it shows that there are 3 possible reading frames, out of which only one reading frame is the correct one. And this has to be identified that which is the correct reading frame. So the frame that is in a frame with the AUG codon is the correct reading frame. So how do we identify the correct reading frame? That is also to do with the initiation process. So here we can see the 30S ribosome, which has 3 different sites. The P site, which stands for peptidyl transfer RNA binding site. The A site, that stands for amino acid transfer RNA binding site. And the E site, which is also known as the exit site, wherein the empty transfer RNA exits from. This is a messenger RNA sequence that is initially placed at the correct location on the ribosomes. So basically, the ribosomes read the messenger RNA sequence wherein you have specific interaction taking place between the Shine-Dalgano sequence that is present upstream of the start codon, and there is specific base pairing that is taking place between the Shine-Dalgano sequence and the 16S rRNA, which is part of the 30S ribosome. This base pairing ensures that the start codon is being placed at the correct position, that is the ribosomal P site. Also, whatever is the frame of this AUG codon, that is considered as the correct reading frame, or the protein is synthesized according to the frame of the AUG start codon. So 3 different interactions, they basically ensure proper binding of the initiating formaldehyde methionine transfer RNA to the ribosomal P site. First is the codon-anticodon interaction between the formaldehyde methionine transfer RNA and the initiating AUG codon. Second is the interaction between Shine-Dalgano sequence and the 16S rRNA ribosomal sequence. And third is the binding interaction between formaldehyde methionine transfer RNA and the ribosomal P site. Coming to the initiation stage in prokaryotes, first there is dissociation between the 30S and the 50S subunits of the 70S ribosome. So they tend to reassociate again with each other. So first they need to be in a deassociated form so as to begin the process. So here uh, initiation factors 1 and 3 are involved, which interact with the 30S subunit and prevent its reassociation with the 50S subunit by bringing about conformational change in the 30S subunit. Then the 30S subunit reads the messenger RNA molecule through specific interaction between the Shine-Dalgano sequence and the 16S uh, rRNA sequence of the 30S ribosome. The AUG codon is placed precisely at the ribosomal P site. Next is the entry of uh, the initiating formilated methionine amino acid that is already forming a complex with the uh, initiating transfer RNA. This complex will form a larger complex by interacting with initiation factor 2 GTP complex. Now, by codon-anticodon base pairing, the transfer RNA will bind specifically to the AUG codon that is present at the ribosomal P site. After this, there is entry of the 50S uh, subunit of ribosome. Simultaneously, there is hydrolysis of GTP, releasing GDP and inorganic phosphate, and release of all three initiating factors 1, 2, and 3. Um, this leads to formation of a functional 70S ribosome, which contains the 30S subunit, 50S subunit, the messenger RNA molecule present in proper location such that the AUG start codon is present at the ribosomal P site. The initiating transfer RNA carrying formilated methionine present at the ribosomal P site by codon-anticodon interaction. This marks the end of the initiation stage, and then the protein synthesis process can now enter into elongation.